So a very general definition is that the heritability is the proportion of a population's phenotypic variance that is attributable to genetic variation. Heritability does not indicate the degree to which a trait is genetic. It measures the proportion of the phenotypic variance that is the result of genetic factors. Now, there are two slightly different definitions of heritability. So you've got broad sense heritability and narrow sense heritability. Let's start with broad sense. So broad sense heritability is the proportion of a population's phenotypic variation that is attributable to genetic variation. And it's equal to capital H squared equals the variance due to genetic factors over the total phenotypic variance. So to estimate it, you would calculate from genetically identical lines, make homozygous lines, and then cross to get various heterozygotes that are genetically identical. And then since the phenotypic variance equals the sum of the genetic variance and the environmental variance, and because you've got genetically identical population, then your variation that's caused by genetics is zero. Hence, the total phenotypic variance is caused by the environment. Now you can calculate the total genetic variance by taking the total phenotypic minus the environmental. So that's one way of doing it. You can also calculate from the differences in genetic correlations between different types of related individuals. If H squared is much greater than 0, then genetic differences influence the phenotypic variation. If H2 equals 0, then in the population studied and in the environment studied, there is no effect of genetic variation on phenotypic variation. Now, coming on to narrow sense heritability, the proportion of a population's phenotypic variation that is attributable to additive genetic variation. That's the main difference. Broad sense was total genetic variation, whereas narrow sense is additive genetic variation only. So little h squared equals additive variance over total phenotypic variance. So usually this is calculated from parent-offspring regression plots where y equals bx plus a and b equals the narrow sense heritability, or little h squared. So if b equals 1, then VA is the only component that influences variation. If b equals 0, then the additive variance does not influence variation. And if it's anywhere in between, then you've got the additive variance and other components which influence the variation. This is important for breeding programs because the calculations simply look at the likelihood of being able to select for an observable phenotype based on the phenotype of the parents and the heritability of the trait. So the selection response is the difference in a trait between the selected individual's progeny, the average, and the average of the previous generation. So little h squared times the selection differential, and the selection differential is the difference between the selected parents and the unselected mean. When selection is imposed upon a phenotype, the phenotype will change from one generation to the next, provided that there is still genetic variation in the population that affects the trait of interest. Heritability and selection. For a population to experience natural selection, two conditions must be met. The first is that individuals must vary from one another for a particular trait, and the second is that an individual's survival and reproductive success must be affected by which of the traits it possesses. The heritability estimate is specific to the population and environments you are analysing. So just remember, going back to the norms of reaction, it's not you cannot draw conclusions for one environment and then say that that is definitely the case in another environment. Some traits will be well adapted to a given environment and some will not. For instance, Darwin's finches with larger beaks tend to survive periods of drought more successfully than those with smaller beaks. Note that selection happens within a generation. However, natural selection says nothing about what happens to beak size in subsequent generations. So 3 criteria for evolution by natural selection. So the individuals in a population must vary from one another. Survival and reproduction must be affected by that variation. And thirdly, the variation must be heritable. So I've now added on the third clause. A fundamental equation quantifies the evolutionary response selection. So see Falconer 1989. The formula is simple yet wonderfully useful and is the following. Big R equals big S times little h squared, where R stands for the evolutionary response to selection, a measure of how natural selection causes a population to evolve. S is the strength of the selection, also known as the selection differential, and little h squared is the heritability, in the narrow sense. In order to understand big R, let's first discuss the concepts of selection, selection differential, and heritability. Selection and the strength of natural selection occurs whenever survivorship or reproductive success is non-random with respect to a particular trait. Selection can be either directional, stabilizing, or disruptive. Let's look at those 3 types of selection a little bit more in detail. So in directional selection, survivors are at either of the high or the low end of the variation in a trait. For example, in a caterpillar, there would be directional selection for weight of the caterpillars. The fattest caterpillars survived. That's the ones at the extreme high end of the population. In stabilizing selection, individuals with intermediate values survive the best, whilst individuals at both extremes don't survive as well. For example, suppose that small caterpillars did not survive due to insufficient resources to survive pupation, but very large caterpillars did also not survive well because predators such as birds were better able to see and eat them. Then the best survivors would be the caterpillars with an intermediate size. In this case, the caterpillar population is experiencing stabilizing selection. In disruptive selection, individuals in the population with either high or low extremes for a trait survive better than individuals with an intermediate size trait, so the exact opposite of the previous one. Suppose that caterpillars varied in their degree of melanism, so pigmentation, with some caterpillars being quite dark, some being quite light, and some caterpillars having an in-between colour. Now, suppose that the caterpillars were found on both the white bark of birch and the darker bark of a walnut tree. The light caterpillars would be protected from predators because they would be hidden on the white bark of the birch tree. The dark caterpillars would be protected on the bark of the walnut tree. However, caterpillars with the intermediate colouring would be visible to predators on both types of tree, so they would not survive as well as either of the extreme colours. How do you measure the impact of natural selection on the population? You could take the mean weight of the population before any individual died, and then compare it to the mean weight of those individuals that survived natural selection. This is what S, the strength of selection or selection differential, measures. The difference in the population's trait before and after natural selection. If S equals 0, then survivors and non-survivors should not differ, don't differ in this trait, and the offspring of the survivors should not differ from the previous generation. The larger the value of S, the more intense the action of natural selection on the population. Heritability, h squared, E. The total variation in the population is thus Vg plus Ve. and little h squared is then equal to Vg over, so Vg is on the numerator, and then on the denominator you've got Vg plus Ve. So we're just rearranging the equation that we had earlier of Vg over Vp, the total phenotypic variation. This is assuming there's no sort of interaction between the genetic and the environmental factors. So theoretically, h squared can only vary between 0 and 1. When variation among individuals in a population is due entirely to differences in environmental conditions, then little h squared equals 0. When the total variation among individuals in a population is due solely to differences in the genotypes of individuals, little h squared equals 1. Quantitative geneticists calculate the heritability in two ways: by manipulating equation 1, which was the R equals S times H. You can solve for heritability as little h squared equals R over S. This is the realized heritability, or heritability defined by the degree to which a trait responds to selection in a population. A second way to solve for heritability is to graph the trait in a set of offspring against the mean of the trait for each of the two parents, called the mid-parent value. The slope of the regression line for such a plot is one way to estimate heritability.